Okay. Hello, good morning, good afternoon, good evening, everyone. Welcome back to our class. Yeah. So again, since we have done with the discussion on digestive system and also with the discussion on excretory system, today we will be discussing about the cell cycle, wherein we are making brand new cells here. When we say brand new cells, these cells are young, these cells are capable of growth, these cells are also capable of reproduction. Okay? So that's what we meant when we say brand new cell. So what is cell cycle? There are a lot of terminologies actually that we need to discuss or we need to find out for us to discuss further about cell cycle or for us to understand about cell cycle. So we are going to label the different images or the different parts as we go. So we have here your first is your chromosome. So this is your chromosome. By the way, what is a chromosome and what do they do? So that's where what we're going to find out. The chromosomes actually are thread-like structures located inside the nucleus of a cell. So when we say chromosomes, they are located inside the nucleus of the cell. Okay? And not only an animal cell, but they are also in plant cells. Right? So each chromosome is made of protein and a single molecule of deoxyribonucleic acid. We have discussed about deoxyribonucleic acid before, right? When we say deoxyribonucleic acid, this is actually the acid that is capable for our traits, for our genetic makeup. So when we say deoxyribonucleic acid class, take note that this one is responsible for the trait that we inherit from our parents. Okay? For the trait that we inherit from our ancestors. Okay? Kung kamukha mo sa Angelina Jolie, then you might be from the ancestry of Angelina Jolie. Okay? Ganon. Kung kamukha mo naman si Albert Einstein, then you might be from the ancestry or genealogy of Albert Einstein. So, ganun lang yun. Alright? So, passed from parents to offspring, and then DNA contains the specific instruction that make each type of living creature unique. Tama po yan. Why? Why is it unique? Why does it make every individual unique? Okay, look at our fingerprints. Why do you think are the reason or is the reason why they are getting the fingerprints of people, right? Okay, because each fingerprint, okay, of or the fingerprint of each individual is very unique. Wala po silang katulad. Okay, wala silang katulad. Okay, kaya kung anong fingerprint mo, hindi pwedeng maging fingerprint yan ng ibang tao. Kung anong fingerprint ko, hindi pwedeng maging fingerprint ni Catherine Bernardo. Hindi pwedeng maging fingerprint ni Liza Soberano. It's because each fingerprint of each individual is very unique. Okay? And the spaces, okay, the spaces in our fingerprints, okay, yung mga paloob, those are what we call the root ridges. Okay, so that's our chromosomes. And what's the function of our chromosomes? What do? They actually play a very important role in cell division. Okay, because they contain the DNA. And the DNA, dyan po nakasalalay yung ating magiging itsura. So without the chromosomes, there are possible mutations. Like for example, magkakaroon tayo ng mutations such as magiging Turner syndrome. Okay? So mga mutation yung mga yan. Crudichat, okay, yung cry of the cat. Ano pa? Actually, madaming mutations yan. Pwedeng magkaroon tayo ng 6 finger dito. Pwede rin tayong magkaroon ng 4 na finger lang. Pwede rin tayong magkaroon ng malaking mata dito and then maliit na mata dito. So they are all actually made up by our DNA. So the DNA decides our traits. So kung anong trait ng parent mo, yun din ang makukuha mong trait eventually. Okay? Understood? Okay. We also have here your centromere. And what's the function of your centromere? Aside from it's in the middle, so actually in real life, centromere are not always located in the middle. It just so happened that this is in the middle. Okay? So centromeres help to keep chromosomes properly aligned during complex process of cell division. What happens ba? What happens if the centromere is not there? Okay? So the chromosomes will be separated from each other. Okay? So the chromosomes will not be will be separated from each other, causing them to cause multiple mutations. Okay? So magkakaroon ng halo-halo. Genetic mix-up ang tawag natin doon. Magkakaroon tayo ng madaming halo-halo. And there will come a time that these mutations will actually cause defect on the organism that will arise. Okay, understood? So that's centromere. Okay. We also have here a common representation of our cell cycle, wherein we have taught that cell cycle starts with the parent cell. So this is actually the parent cell. And then this is where the replication of the different parts or the replication of um, the different organelles in the cell. And then eventually, when the different organelles are replicated, they will be divided into two. So that's what we call your cytokinesis. Cyto means cell or cytoplasm, and then kinesis means splitting up. So that's the splitting up of your cytoplasm. Okay, understood? All right. So we also have here your spindle fibers. What's the function of our spindle fiber? Okay, so our spindle fiber plays a very important role in keeping the chromatids or the sister chromatids intact with each other. So kahit medyo nagkahiwalay sila, at least there's still a bond that connects the two of them. So there will come a time that these two will bond again. And aside from that, they keep the sister chromatids away, okay, away from each other, but they are not allowed to mix up with other chromatids. That's why it keeps our body or it keeps our organisms or our offsprings from getting multiple mutations. So that's how important your spindle fibers are. So these spindle fibers are just like a thread. Okay, it's very small, but it's so powerful that it could prevent mutations, it could prevent other damages during the cell division or other defects during the cell division. Okay. So we have here your cell division. So example, this is only an example. We have here separating centrosomes. This is your mitotic spindles, the microtubules, this condensed chromosome. So that's your chromosome. Centromeres. So this is your centromere, the orange one. And then you also have your nuclear envelope. So as you can see, your nuclear envelope is here. And if your nuclear envelope is here, then therefore chromosomes are located inside or outside? Okay, very good. Chromosomes are located inside the nucleus. All right, very good. You're getting good at it. So now let's discuss the interface of our cell division. When we say cell division, it will take a lot of process for you to prepare before you could actually go to the main process. So we have three preparatory processes or three preparatory stages before we can actually have your four main process or four main stages. So we have here your GAP1 first. Okay, so this is G1. So that's GAP1. So in your GAP1, cellular contents, excluding chromosomes, are duplicated. So lahat daw, okay, lahat daw ng cellular contents except chromosomes, they are duplicated. So in your synthesis stage, this is actually the synthesis stage, S. So each of the 46 chromosomes is duplicated by the cell. So kanina sa gap 1, ang na-duplicate lang po are the organelles. But in your synthesis, what's duplicated here is your chromosomes. Okay, and your chromosomes is 46. Bakit 46? Because that's the number of chromosomes in a person. Okay? Kaya hindi pwedeng mag-asawa or mag-mate ang tao at unggoy. It's because they have different numbers of chromosomes. As well as the issue in Robinson na yung tao nanganak ng ahas, yung kapatid niya ay ahas. So hindi po yun possible because the number of chromosomes, that's according to biology actually, the number of chromosomes of a person is quite different or is different from the number of person of other organisms. Okay, and the number of chromosome of other organisms. Okay, so do not believe on those hopes. So we move on to gap 2. That gap 2, the cell double checks. So hindi lang si teacher na double check, hindi lang din kayo, but also the cell. So the cell double checks and duplicates the duplicated chromosomes for errors and making any needed repair. Imagine if we're going to shift or if we're going to skip gap 2, it means the cell does not need to double check the duplicated chromosomes. Anong mangyayari? What happens is we will undergo mutation or we will more likely develop mutation. Why? Because our cells or our body can make damage all the time. Actually, hindi perfect ang katawan natin eh. Okay? It can create a lot of damage all the time. However, it's very important that we or our body double checks the chromosomes. It's for us to prevent mutation para hindi tayo nagmumukhang may Turner syndrome, may cri du chat, and so on and so forth. So madaming syndromes actually. Alright? So I hope you understood my point. Okay, so that's gap 2. And then after gap 2, we have meiosis. Okay, mitosis. So mitosis actually, it's a cell division on your somatic cells. Okay, somatic. And then when we say meiosis, that's your cell division on your sex cells. Okay, so don't forget that mitosis, that's your cell division on somatic cell. And then meiosis, that's your cell division on sex cells. Okay, very good. Okay, that's nice. Okay, so we have here, it is always the life of a eukaryotic cell. The cells grow, make new copies, and divide. So very important po na mag-grow ang cell natin, magkaroon ng bagong copies, and mag-divide because it's one of the representation that we are alive. And what are the other representations? We reproduce, okay? We grow, we undergo biological processes. So those are the common symptoms or the common reasons on why we really have to undergo cell division. Okay, so it happens in our somatic cells or body cells in order to get the same DNA inside each cell. So your reproductive cells do something different. So later on in this topic, we are going to talk about the cell division on your reproductive cells or your sex cells or your gametes. Okay? So parent cells are diploid and they make two daughter cells that are also diploid. When we say diploid, that means each chromosome, okay, two of each chromosome is, okay, 23, tapos dalawa sila, so magiging 46. Phases of the cell cycle, ibig sabihin, you have your IPMAT-C. Okay, tandaan nyo lang yan, IPMAT-C. I feed in the mat. C, okay. I feed on the mat. C. Okay, so if MATC, we're in I stands for interface, so that's our GAP1, synthesis, and GAP2. And then P means prophase, the first stage on mitosis. We also have here meiosis, okay? Metaphase, sorry. And then we also have here your anaphase and then your telophase. And then the last one is your cytokinesis. Okay, so let's talk about the largest phase. Actually, the largest phase, which is 95% of growth, occurs in your interface. So this is the time between cell divisions. The cell is growing, okay, copying its DNA and preparing for division. And then the copying of DNA is called synthesis or replication. And then we also have three stages. We have G1 or GAP1, synthesis, and then GAP2. So later on, we are going to discuss about the different processes that needs to be undergone in GAP1, GAP2, and then in synthesis. All right? So we have here your GAP1. Actually, in your GAP1, growth of cells inside and the insides and the development, differentiation, the cell is told to what become. So in your GAP1, lumalaki yan. Bakit lumalaki? It's because the organelles are replicating. Okay? Nagre-replicate yung vacuole niya, nagre-replicate yung lysosome niya. Okay. So, lumalaki yan kasi dumadami din yung laman niya. And then we also have here synthesis of DNA and it's also called replication. So, if we're going to look at it, the DNA double helix is unzipped. Okay, yung itsura ng unzip, no? Completely by an enzyme called helicase. When we say ASE, that's actually an enzyme. And the function of this enzyme naman, it is to unzip or you decode the DNA. So one DNA nucleotide at a time is added to both sides of the DNA strand. So A, it means partner niya si T. And then si C, partner niya si G. Ano bang mga meaning ng A, T, C, G natin? When we say A, okay, that's okay. So when we say A, that's actually your, okay, when we say A, ano siya? Okay, when we say A, that's actually your, okay? So when we say A, that's actually your adenine. So ang adenine po, partner niya si T. So T stands for thymine. So they are actually the nucleotide base pairs. Okay, we also have C, that's cytosine. Okay, and ang partner niya si G, guanine. So again, A, adenine, thymine, cytosine, guanine. So adenine, sa kanya si thymine. Si cytosine, sa kanya si guanine. Okay, so with the help of another enzyme called DNA polymerase. So the DNA polymerase actually is the one that is responsible for the bonding and for the separation of these nucleotide base pairs. So the two nucleus is left, okay, with the nucleus is left with two exact copies of all the chromosomes of DNA. So let's check replication. Okay. So later. So the G2 actually, the cell prepares for cell division and checks for error. So we have discussed that a while back. So this is actually cell division, ay replication rather. So nakikita niyo naman dito si C, partner niya si G. Si T, partner niya si A. So this is actually, okay, the unzipping. So dito na papasok si helicase. So kapag ito na si helicase, mag-unzip na yan. Okay, mag-unzip na yan. And this one is responsible, okay, the DNA polymerase naman para mag-zip sila ulit at some point of time. Alright, so that's it. So that's your cell cycle start. Okay, G1 or GAP1, wherein nag-duplicate or nagre-replicate yung mga organelles natin, and then the synthesis. So ang synthesis natin, it's actually the part wherein our chromosomes are duplicated, and then we also have your gap2, the duplicated chromosomes, and then we have now your mitosis. So that's all for today. I hope that you're going to watch this video. If you have questions, please leave them in the comment box below. Or you can just directly email me at at dianarose.flora@lorma.edu. Or you can contact me in Messenger, Diana Flora, or you can just send your queries below. Okay, goodbye everyone. I'll see you again soon.